Children, good afternoon and welcome back. So the next topic is about cloning vectors. You know what are vectors? Already you know. But when the cloning vectors, what are these clones now? What is cloning or what is clones? When you incorporate this recombinant DNA into the yeast or bacterial cell, it will replicate, right? And form same type of the recombinant DNA, which is called as clones, right? Okay. Now, when the cloning vectors are multiplied in the host, the linked piece of DNA is also multiplied to the number equal to the copy number of the vectors. Remember what is copy number? The ORI will control the copy number of the plasmid. Okay. Right. Now this is what are the features required for cloning into a vector. So here children, this one, what the plasmid shown here is an artificially synthesized plasmid. Okay, now what are the features it has, or what are the features a cloning vector should have, right? So it should have, first one, an origin of replication. Okay, and second thing is it should have a selectable marker, and the third thing is it should have cloning sites. Okay, these are the three main features of a cloning vector. Now what is this origin? Okay, let me talk about here. So these are, what are these restriction sites or cloning sites? These cloning sites are also called as restriction sites. Restriction sites. Okay, which are these restriction sites of this cloning vector, and what is the name of this vector, children? It is PBR322. PBR322 is the name of this vector, cloning vector. Okay, but restriction sites are, you can see in this figure, which are they? This is HIND2, HIND3, PVU1, PST1, BAMH1, SAL1, PVU3, 2, everything. All these are the restriction sites. What is restriction sites? It is the place where the restriction enzymes can act and cut the DNA. At this point, this is the restriction site of this restriction endonucleases. Okay, and it has ORI. ORI, where is ORI? This place is the ORI. Okay, ORI. And these are the selectable markers. Which are the selectable markers? Which is the tetracycline resistant gene in this site and the ampicillin resistant gene. Tetracycline, TET, superscript R, which means it is tetracycline resistant gene. The other one is ampicillin. R means resistant gene. What are these tetracycline and ampicillin? These are antibiotics. And usually the bacteria, you know, they are not resistant to these antibiotics. Usually they are not resistant. But this cloning vector do possess these two genes, and they are considered as the selectable markers. And there is one more thing which is called as, which is a place called as ROP. ROP. Here, ROP. Okay, ROP is the gene which codes for the proteins involved in the replication of plasmid. Proteins are needed for replication, right? Adine code cheyyunnathaanu this ROP region. ROP is the codes for replication of proteins which are needed for the replication of plasmid. Okay, now we'll go into each of these things. What is the first thing is the origin of replication, ORI, origin of replication. So it is, what does this region indicate? It is a sequence where the replication start. Okay, So in bacterial, the plasmid is a round structure of DNA, and they don't have a 3' and 5' end. Okay, and there is a place called as ORI, okay, which is called as origin of replication. A piece of DNA linked to this ORI can replicate within the host cells. And you also know that it is this ORI region which controls the copy number, which I just told now, which is involved in the copy number of this plasmid. Sometimes in a cell or per cell there may be one plasmid only. There may be 10 plasmids in some other E. coli, in maybe 200 or so, maybe in some other bacteria. So which is favorable for us? Which bacteria is favorable for us to use for recombinant? The one with one recombinant, the bacterial cell with one plasmid, or 10 plasmid, or 100 plasmid? Definitely 100 plasmid, right? Okay, which because the ORI is the one which controls this number. Clear, children? Okay. And the second thing is selectable marker. So I told the selectable, which are considered as selectable markers. There are other genes also, children, on all these sites, PVS1, PVU21, all these are restriction sites which contains markers, of course. Markers are genes, but we select ampicillin and tetracycline resistant because the bacteria are already not resistant to these two antibiotics. So we can use this as a selectable marker. Okay, selectable markers helps to select transformance from non-transformance. I'll tell you what is transformant and non-transformant, children. It is if a piece of DNA is introduced in a host bacterium, it is called as, it gets transformed, right? When it is incorporated into the DNA, into the plasmid, we can call the transformation has happened. Such bacteria is called as transformed. If the transformation does not happen, if our recombination technique has failed, it has not just failed to get incorporated into the DNA or plasmid of that bacteria, then we can say it has not got transformed. That transform aitla, recombinant DNA aitla. Okay, so we can call them as the non-transformants. Okay, so this selectable markers. What is the use of selectable markers? It helps us to differentiate between transformants and non-transformants. Clear, children? Okay, right. Next is the cloning sites. Okay, if I ask you which are the cloning sites in this vector PBR 3 to 22 means, what will you say? You can say HIND3, PVU1, PST1, PVU3, BAMH1, SAL1, all these are the cloning sites. So these are the cloning sites, children. So this is the cloning site where the restriction enzyme will act and cut this part. Okay, and in this site, which selectable marker is there? There is ampicillin resistant gene. Okay, if this is the cloning site you are using for making recombinant DNA, what is the selectable marker present there? We have tetracycline resistant gene. Okay, now you should know, if this part, this is by using restriction enzyme, this part of the DNA is cut and this cloning site is removed, or restriction site is removed, and a new recombinant DNA is inserted here, suppose, right? So that means what? This plasmid PBR 322 will lose the tetracycline resistant gene. Am I right? Okay, then it is called as insertional inactivation. What is that? Insertional inactivation. I'll tell this side also. Suppose I'm using a restriction enzyme here at this site, this cloning site. Okay, and I'm cutting this part and I'm introducing a recombinant DNA into this portion. using the same restriction enzyme there also. So here this site has this ampicillin resistant gene, right? And this ampicillin resistant property of this PBR322 is lost. Am I right? Am I correct? Okay. So this thing is called as insertional inactivation. Clear, children? Okay. Now children, what will happen if all the restriction sites are used and it is cut using restriction enzyme? What will happen? This PBR322 will become into pieces, and our aim is not accomplished. Clear? So only one or two of the restriction sites or cloning sites can be used for recombinant technology. Clear? More than one recognition sites will generate what? Several fragments. It complicates the gene cloning. Clear, children? Okay. So ligation, what is ligation? Pasting of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. For example, if ligation of a foreign DNA at BAMH1 site of tetracycline resistant gene, as a result, recombinant DNA അല്ലെങ്കിൽ recombinant plasmid is formed. If ligation does not occur, that means it is called as a non-transform, and it is not transformed at all. So we can call it as non-recombinant plasmid. Clear, children? Children, when you incorporate a DNA into the plasmid, what is happening? Insertional inactivation is happening. Okay, that means the recombinant plasmid will lose tetracycline or ampicillin resistance nature. Okay, when the plasmids are introduced into E. coli, 3 types of cells are obtained. One is the transformant with the recombinant DNA. The other is non-transformants, which do not have the recombinant DNA. And also the third type of cells formed is transformants with the non-recombinant plasmid. Or 2 types of cells is formed, that you remember. Transformants is formed with recombinant DNA and non-transformants is formed. At least that you remember. Clear? Now, how do we know? How do we know whether it has got transformed or not transformed? How do we understand whether it has got incorporated into it or not? Suppose we have incorporated a foreign gene into BAMH1 site, which means there in that site, what is there? What gene is there? Tetracycline resistant gene is there. Okay, now it has become lost, so which is called as insertional inactivation. Clear? Okay, now we have to find out whether it has got transformed or non-transformed, or it just not got into it. And how to do that? We have to allow the bacteria to replicate a few generations. Okay, it takes nearly 28 minutes to replicate, you know that. Yes. So that is